Hello, you're welcome to today's class. We are going to talk about the astrocyte metabolic pathways and neurological disorder. Okay, the astrocyte play a central role in brain's metabolic homeostasis, regulating both energy and redox balance. In the event of neurological injury, astrocytes can be activated by or in response to insult. So there's what we call reactive astrogliosis. This is a common pathological feature in many neurological disorders and may play a role in neuropathological pathological progression. So dysfunction of astrocytes and regulatory pathways, including proteins, ion channels, protein synthesis, may also lead to development of neurological diseases. For instance, impairment of astrocyte glutamate uptake and metabolic function can lead to neuronal excitotoxicity and neurodegeneration. Um, the neurological disorders such as the depression, dementia, the Alzheimer's disease, the epilepsy all show impaired astrocytic metabolism. Now, certain neurological imbalance has been associated with reduced glial densities in different brain regions. So changes in this glial distribution may cause a shift in the brain's metabolism. So a lot of research has been done, and most of them suggested that there is a strong correlation between changes in brain's metabolic functionality and neurological disorders. So most recent research, though, has revealed a strong connection between associated functionality and neurological disorders. Most of them, most recent, alteration in associative function, particularly metabolic function, may be a key reason for worsening of this neurological disease. Now, let's take a look at this diagram. We are going to explain the impact of associated metabolic pathways on neurological disorder like the depression, or the Alzheimer's disease, and the epilepsy. Now, looking at the depression, let's start with the depression. Here we call it the major depressive disorder, the MDD. Actually, this depression disorder is a neurological condition caused by a chronic exposure to stress, and it is also characterized by the loss of motivation, what else? Impaired social interaction, communication, and pervasive sadness. So, patients in this situation, that is the depression disorder, were, according to research, were found to exhibit reduced blood flow in glucose metabolism in the brain. So the MDD patient have also been found to have impaired tricarboxylic acid cycle functionality, and which decreases energy production and may lead to exacerbation of depression-like symptoms. Yes, a decrease in the depression disorder, a decrease in number of astrocytes may lead to an imbalance of neurotransmission, synaptic connectivity, and general metabolism as well. So looking at this diagram, in depression, metabolic pathway are impaired in astrocytes, such as the EET. You have the EET here. They are impaired. The EET is the epoxy eicosapentaenoic acid signaling pathway. We have the PPP. Um, we have the PPP. We have the TCA cycle, and we have the increase in the ROS formation. So these changes in astrocyte leads to a decrease in dopamine, ATP, glutamate, the astrocyte itself, the serotonin, and also majorly ROS. but in this case, ROS is being what increased. Now, looking at the second, we have the associated metabolic pathway in Alzheimer's disease. Astrocyte's metabolic pathway provide, of course, we know energy to neurons for various functions through neuron modulation, such as memory, motor, and I think the cognitive functions. And this Alzheimer's disease is a neurodegenerative disorder that is characterized by progressive cognitive decline, as well as we also have loss of memory and dementia. So there are many theorized causes of this Alzheimer's disease. So one of the main causes being metabolic dysfunction. So significant metabolic coupling is present between astrocyte and neurons, especially during the synaptic activities. So the onset stages of this Alzheimer's disease, according to research in a mouse model, using mouse model, there was a progressive astrocytic atrophy with increased GFAP staining in the cortex and the hippocampus of the brain. So looking at this diagram, so metabolic pathways signaling were impaired in astrocytes, such as the glutamate uptake pathway, the glycolysis pathway, the TCA cycle, and there was even reduction in insulin resistance. But in all, there was increase in what reactive oxygen species. Now, talking about the epilepsy, let's target the astrocyte metabolic pathway in epilepsy. The astrocytic regulation of metabolic function is paramount in the role it plays when it comes to epilepsy. Epilepsy is caused by the imbalance of the excitatory and inhibitory neurons in the brain, which may be a cause of metabolic dysfunction. So astrocytes can participate in neurotransmission by regulating ion concentration and also the neurotransmitter. So the epilepsy in epilepsy, astrocytes adopt a reactive morphology, uh, become uncoupled and it also lose domain organization. So these changes can have a variety of influence on functionality. These morphological changes may lead to dysfunction in glutamate clearance, changes to associated functionality in epileptic condition may lead to epileptic symptoms, may increase epileptic symptoms. During epilepsy, the increase in potassium flux, for example, may result from ion channel dysregulation. So looking at this diagram here, the pathway involved in epilepsy in astrocytes, such as the metabolism of glutamate, the synthesis of GABA, glycolysis, lactate, glycogen metabolism, they were all impaired, leading to accumulation of glutamate, lactate, and glycogen, and the loss of GABA and glucose. Thank you, and do not forget to subscribe to this channel.